Bismillah, Assalamualaikum warahmatullah. You're listening to microbiology lessons on microbial growth and control, part 3. In the last episode, we left at physical antimicrobials. So in this last part of the topic, we're going to continue with chemical antimicrobials and antimicrobial therapy, and then we're going to finish it off with antimicrobial resistance. One basic but crucial concept when we are talking about chemical antimicrobials is the difference between bactericidal agents versus bacteriostatic agents. Bactericidal agents are chemical agents that kill bacteria outright. Bacteriostatic, on the other hand, only inhibits the bacteria. The distinction between these two is not necessarily binary, by the way. For example, you can have the same chemical agents that are bactericidal at higher doses and bacteriostatic at lower doses. There are multiple ways you can evaluate the effectiveness of your chemical agents. For example, you can use what's called phenol coefficient. Here, you use numbers that express how good a chemical agent is relative to phenol. So if your lab protocol uses ethyl alcohol and you look up the literature online and you find its phenol coefficient is 6.3, that means you need 6.3 times less concentration of ethyl alcohol than phenol to kill that microorganism. So fine. And then you discovered another chemical agent and you want to replace ethyl alcohol with it because it's cheaper or whatever, safer. So if you can find out its phenol coefficient, you can compare its performance to ethyl alcohol. You might find that it's doing well against Staph aureus, so you can replace it to save cost. But you can't use it against Salmonella because you would need too much of it in order to achieve the same effectiveness. So that's an example of how you can use phenol coefficient. Another method is disk diffusion. There are different ways of doing this. For instance, you take a paper disc, you dip it in your antibiotic, and then you place it on the surface of your agar plate. The agar plate, of course, has microorganisms inoculated on it. After you incubate the plate, you're going to see a clear area around the disc. So that indicates the bacteria had been killed. So here you can infer that the wider the clear zones are, the more effective your antibiotic is against that particular bacterium. And then you can compare them. When choosing chemical agents for your lab work, you want to think about a couple of factors. You consider its efficiency, meaning how quickly and the minimum dose it needs to get the job done. Its stability around organics, meaning can it do its job when you apply it to organic materials? How many targets can it be effective against? Is it damaging to other organisms or materials that you use in your research? Also, other practical factors such as how much it costs, how difficult it is or how easy it is to prepare, and how available are the ingredients if you want to prep the solution in-house. And is it sensorially non-offensive? For instance, does it smell okay to be used consistently? So those are factors for choosing the right chemical agents. Chemical agents work through a number of mechanisms. So it can work on the microbial proteins, for example, by causing denaturation of the tertiary structure, or hydrolysis, where it breaks the primary structure of your protein using water molecules, or it can damage the protein by oxidizing the chemical linkages in it. When you apply chemical agents to bacteria, when it comes to denaturation, you can have permanent denaturation or temporary denaturation. When your chemical agent causes permanent denaturation, your agent can be considered bactericidal. So one helpful visual to understand this is when you fry an egg, you cannot unfry it. The protein structure of the egg is permanently changed. Some bacteriostatic agents can only cause impermanent denaturation. Once the agents go away, the bacterial protein can reconfigure back to its original form. An analogy for this is when you warm milk, the protein structure of the milk changes. But later, when you cool it down, the protein structure will go back to its original configuration. So that's the difference between permanent and impermanent denaturation. Another mechanism is by targeting the microbial membrane. Bacterial membranes usually have lipids in them, so you can apply a surfactant compound to target those lipids. What happens is the long hydrocarbon end of the surfactant will penetrate the greasy, oily structure. And then at the end of the surfactant, you have your carboxyl group, and that carboxyl group will attract water molecules. The water molecules will then aggregate around the grease, and that will be enough to remove it from the surface. So this is what happens, for example, when you wash your oily plates with soap. So you can find more examples of how various chemical agents work in most microbiology textbooks and online resources. And some of these antimicrobial agents can attack multiple targets at once. For example, phenols can attack the membrane by disrupting the membrane structure, and at the same time, it also denatures the proteins and inactivates the target bacterial enzymes. We use chemical agents as part of chemotherapy. I know, before we study microbiology, when we hear chemotherapy, we think about cancer chemotherapy. But technically, chemotherapy is any type of therapy that uses chemical agents. And when we use chemotherapeutic agents, we consider a number of properties. First is the selective toxicity. We need to determine the dose that is more toxic to the target microorganisms, but less to our own cells. We can use the chemotherapeutic index to compare those chemical agents. This index takes the maximum dose that our body can tolerate, and then we divide it by the minimum dose needed to kill the infection. So the larger the index value, the more desirable and the better a chemotherapeutic agent is. Like if the index value is 2, it means your body can tolerate twice the amount needed to kill the infection, and that's good. The second property is the antimicrobial activity spectrum of the agent. So you can have a broad spectrum of antibiotics like tetracyclines, which can be effective for a wide range of microorganisms. And you can also have narrow spectrum agents like isoniazid, which can be specific to just mycobacteria. Ideally, you want to go for the narrow spectrum antibiotic first, so you don't unintentionally kill off friendly microbiota in your body. We can identify at least 5 modes of actions for antibiotics. The first one is the inhibition of cell wall synthesis. This is for antibiotics like penicillin. The second one is the disruption of membrane function, so carried out by polymyxin. Third are the antibiotics that inhibit protein synthesis, like tetracycline. You also have antibiotics that inhibit nucleic acid synthesis, like rifamycin. And number 5, some antibiotics act as antimetabolites. What I mean by antimetabolites is this. Many microorganisms need a metabolite called PABA, P-A-B-A. They need PABA to make folic acids. Folic acids are vital for downstream synthesis like nucleic acid synthesis. So what we do is give the patients antimetabolites that look like PABA, for example, sulfanilamide or PAS. Now, when the bacterial enzymes try to bind to PABA, because of the structural similarity, the enzyme will as likely bind to the antimetabolites instead. So that disrupts their metabolic pathways, either slowing down their downstream synthesis or in some cases even stopping the pathway altogether. And the fourth consideration is the side effects of the chemotherapy. We have to look at the toxicity of the chemical agents or whether they can cause allergy, meaning whether the treatment can trigger unintended immune reactions, and finally, whether the chemotherapy can disrupt the microflora we have in our body. So in many chemotherapeutic research, the ideal goal is to discover agents that have selective toxicity, where they are effective against infections but safe for the patients. We want the agent to be soluble and stable in the body, meaning that our body doesn't destroy the medicine before it does its job, and you want the medicine to be affordable and have a long shelf life to make it economically viable. And finally, the agent must have a slow resistance acquisition. So antimicrobial resistance happens when your microorganisms survive the antibiotics that were effective before. So you need higher and higher doses. They can acquire resistance in two ways. Firstly, through chromosomal resistance, meaning the genes in their chromosomes mutate and give them that resistance. Secondly, they can get extra chromosomal resistance, which means they get it from all plasmids that are outside of their chromosome. And once the microorganisms acquire resistance, they can defend themselves using the same tactic against different antibiotics. For example, the patient gets penicillin. It has beta-lactam. The bacteria can develop beta-lactamase, and that inactivates penicillin. And it doesn't end there, because now all other antibiotics that have beta-lactam, the bacteria can resist it. Cephalosporins, carbapenems. So this is called cross-resistance. The bacterial resistance is now crossing beyond a single drug. It is almost impossible to stop antimicrobial resistance, but we can do things to limit it. We can use combinations of behavioral and chemical approaches. For instance, when we use antibiotics, we should make it a point to finish the full course of treatment, because when we take antibiotics, the bacteria that are highly sensitive to that particular drug will die off first. And then the intermediately sensitive organisms will start to die. At this point, we may feel better, but we still want to keep taking the antibiotics so that even the highly resistant microorganisms will die as well. Otherwise, if we stop taking antibiotics prematurely, the highly resistant microorganisms will stay around and replicate and spread to other people, and that creates an antimicrobial resistant infection. For the chemical approach, we can deploy what's referred to as double antibiotic therapy. So it works like this. If we do single antibiotic therapy, such as beta-lactam only, the bacteria might develop beta-lactamase. So that can break our beta-lactam, and that inactivates our antibiotics. But when we use double antibiotic therapy, we combine the beta-lactam with quinolone. If the bacteria launch beta-lactamase, it breaks the beta-lactam and releases quinolone, and that quinolone kills the resistant bacteria. And when we have non-resistant bacteria, they still will get killed by the beta-lactam itself. And because they didn't break the beta-lactam, the quinolone remains inactive. And that stops the development of antimicrobial resistance against quinolone. But despite those approaches, the microorganisms can still resist, and they use multiple mechanisms for that resistance. It includes target alteration, where the bacteria change the shape of the protein that is targeted by the antibiotic so that the antibiotic cannot bind to it anymore. They can also change the membrane permeability, like the membrane transport system, for example. So that alteration blocks the antibiotics from entering the bacterial cell through the membrane. Another mechanism is when bacteria develop enzyme activity that specifically inactivate the antibiotics. We've seen this before. Penicillin needs beta-lactam to kill the bacteria, so the bacteria develop beta-lactamase that neutralizes the beta-lactam. Bacteria can also modify enzyme activity. For instance, remember I just talked about how we can use sulfur drugs to mimic PABA? We wanted to confuse the enzyme. We wanted the enzyme to attach to the antimetabolite instead of PABA. So, to resist this tactic, bacteria can modify their enzyme to increase affinity towards PABA. They will know that, hang on, this is not PABA. I'm not going to bind to this. I will bind to the real PABA. So, our metabolite treatment doesn't work anymore. Or, this one is cool, the bacteria can develop new metabolic capabilities. For example, they need PABA to synthesize folic acids, right? So when we send in antimetabolites, that stops PABA from binding. When we see that, we'll be like, yay, you can't use PABA, we win. But the bacteria will go, actually, I only need PABA to make folic acids. I need the folic acids, not the PABA itself. So now I'm going to develop a new pathway, a new metabolic capability to capture environmental folic acids directly. So the war goes on. Over time, that war requires us to develop multi-line drugs, one after another after another. A clear illustration of this is gonorrhea. So this is one of many sexually transmitted infections that people get when they sleep around with multiple partners. So they get infected, they see doctors, the doctors give them sulfonamide, they feel better, and then they sleep around again, or their infected sexual partners sleep around again. They get infected, they see doctors, the doctors give them sulfonamide again, and now it doesn't work. So the doctors give them penicillin. That works. They feel better. They sleep around again. They get infected. The doctors give them penicillin. Now it doesn't work. So the doctors give them spectinomycin. That works. They feel better. They sleep around again. They get infected. The doctors give them spectinomycin. And now it doesn't work. And it goes on and on. So patients with this disease get a 5-fold increase in HIV transmission, which attacks their immune system. They're likely going to get sick with something else. They also can get infertile. They can't have children. Or when they do get pregnant, say the husband sleeps around and then brings back the infection to his wife, the wife is more likely to have an ectopic pregnancy, which means the baby is attached outside of the uterus. So it can cause bleeding and the wife dies. Or the infection gets to the baby. Yes, the husband sleeps around and then he infects the wife and the wife infects the baby, causing neonatal eye infection, and the baby becomes blind. So yeah, you can choose that lifestyle, or you can choose a belief system that disciplines your sexual interaction, one that encourages loyalty to your wife or husband. Your life will be better off by being a disciplined, loyal person. All right, so that's a good place to end. Talk to you in the next one. Barakallahu li wa lakum. Wassalamu alaikum wa rahmatullah.